Okay. So before we get going, I just want to mention that, Jim Estes gave us a class in ecology, and he has organized another series of lectures for January for an online only course. And it's going to be about the ecology of disease. And he's lined up a series of really super research people from all around the country. So this is going to be a really international event. So uh, watch for your Oli newsletter. You'll get the timing on it, but it's basically going to be 4 weeks in January. So that's got a little ways to go. All right, so let's get started. So our speaker today is Josh Arribere. And Josh, do I remember right? You grew up in San Jose, isn't that?
Yeah, San Jose Gilroy, if you know where that is.
San Jose Gilroy. So if you want to look at a stellar CV in terms of training, it would be hard to beat Josh. So Josh got a bachelor's degree at Berkeley. Then he went on to get a PhD at MIT, and then did a postdoctoral fellowship at Stanford University. And just to make sure he had the best people in the best places, his mentor at Stanford was Andy Fire, who won the Nobel Prize in 2008. So we were quite pleased to be able to attract Josh to a faculty position at Santa Cruz. And I should say that despite all of this, Josh is just a regular guy. All right, and he and I have shared a lab for a number of years. Just one other tidbit to add in here, just to keep Josh on his toes. He's one of the many one of the many power couples up at UCSC, since Josh's wife is an internal medicine doctor at Stanford University. I want to say that the faculty at the university are all really pretty stressed out these days and under a lot of pressure, because they're trying to teach online. They're trying to keep their labs going. There are provisions to do that, but under highly restricted rules. They're trying to keep the research going. And then, of course, many of them, have kids at home, so they're being primary school teachers at the same time. So we really appreciate the fact that Josh has taken out the time to talk to us this morning. So Josh, it's all yours.
Thank you. Thank you, Barry. Can everyone hear me?
Yes.
All right. I have a second screen up, so if you want to interrupt me at any point, you're welcome to unmute yourself, or you can just do this in front of the camera and I'll stop and call on you. So as Barry said, I have a lab up here at Santa Cruz. I started at Santa Cruz in 2017 with my own lab. And we've been very fortunate to get some really solid PhD students and techs and undergrads during that time that have contributed to some of the work that I'm going to show you today. I just like starting out with this picture because I think it's pretty. I'll give you a little bit more information on what this picture shows in a couple slides. This is an artist's depiction of the inside of a cell.
It's not coming up yet, Josh.
Uh-oh.
The screen share.
What can you see?
I'm seeing the gallery view.
Oh, okay. Let's fix that then. How about that?
That's it. Yep. Good.
Perfect. And I can still see everyone. All right. So this is an artist's depiction of the inside of the cell. I like showing this just because I think it's visually stunning and pretty. And the more you stare at it, actually, a lot of these molecules are based on the actual three-dimensional structure of these molecules that we now know. And so it's a fairly accurate picture as far as the crowdedness and the number of molecules that are inside of our cells. Oh. All right. So that was the slide that I thought we were all staring at for a while, but apparently it was just me. So I wanted to start out with a slide which is shamelessly stolen from Barry's introduction, which is on the size and scale of biology. So each one of us is made up of trillions of cells, hundreds of trillions of cells. And each one of those cells is made up of thousands, tens of thousands, hundreds of thousands of molecules, each one of which has thousands of atoms. And the ability of the cell to replicate is a huge feat of biosynthesis, because each one of those molecules that it has, those hundreds of thousands of molecules, it has to make again so that it can make a perfect copy of itself and divide. And so, for example, in every one of your cells, there is a genome present in 2 copies, and each single copy of that genome contains 3 billion bases, or 3 billion letters. And it copies it every time it wants to divide. It copies that into 2 separate copies. And you can imagine the incredible feat that that is, to copy 1 billion letters and not make many mistakes. But our cells are not perfect at this. If you look at them very closely, you can appreciate that they do in fact make mistakes. And it's a frequency of about 1 in a billion or so, but they do make mistakes. If you look, however, at the prevalence of errors, it's actually much lower than that. And so that gets into an area that is what my lab studies, which is how the cell helps mitigate the consequences of errors and help mitigate the occurrence of errors to try and keep things at a low and tolerable level. And the analogy that we use is quality control in manufacturing. You can imagine if you have an assembly line and you're producing cars, if you're just taking that car and then putting it on the road without checking whether or not it works, you're going to have a disaster a certain frequency of the time. So you have to be checking your products as they come out of the assembly plant and verifying that there is no appreciable amount of errors, because you don't want to be having the issues that emerge from those errors. And so as cars come out of a factory, they undergo rigorous quality control testing to make sure that they do in fact work, that the brake actually works and the gas allows you to accelerate, the steering wheel is hooked up right, and so on and so forth. That's about the extent of my understanding of manufacturing. I've never worked an honest day in my life. So that's quality control as we can imagine it and as we can visualize it. And quality control during molecular biology works very similar, but it's a little bit different. So as Barry told you in the introduction, DNA, which is where your genome is, gets copied into RNA, and then that is used to make proteins. And so you can think of this as the assembly line that's going on in each of your cells all of the time. So your cells are constantly making RNA, they're constantly making protein, and each one of your cells has to make something like tens of thousands of RNA molecules. Each one of them is thousands of letters long. And so there's a huge amount of space of things that it has to do, molecules that it has to make, and it needs to be very careful about the error frequency of this. The twist, though, is that we have the benefits of eyes and brains, and we can think very rationally, and we can test a car. But it's very dark inside of a cell, and there's nothing that has an eye, there's nothing that has a brain. So how do you build a set of rules to ensure that the molecules that you make are accurate and functional, but you can't use your eyes? So everything's blinded. So everything happens by chemical reactions and by molecules binding to other molecules in a very dark place. So that's the sort of challenge of quality control. So you can think of quality control as cars coming out of a factory. That's pretty straightforward to imagine because that's in our world. But in the cells world, everything is done by binding and chemical reactions and kinetics. Any questions so far? I'm not seeing any hands waving, so. Seeing a couple people talking, but I assume they're talking to. All right. And this is the complex that my lab studies a lot. This is one that Barry introduced to you, and this is the ribosome. So the ribosome is a giant machine that takes RNA in and then outputs protein. And these proteins are what make up the bulk of your cells. They help you store memories, for example. They help give your skin elasticity. They help pull cells apart during cell division, and so on and so forth. The proteins really carry out a lot of the molecular functions. So again, Ribosome is what carries out this step of RNA going to protein. And so the cell has a very roundabout set of rules it uses to ensure the quality of what it makes. And I'm going to list some of those rules here. So some of the rules that cells adhere to is that a ribosome has to stop at a certain point. And that point cannot be too early and cannot be too late. And if it is, the cell gets concerned that that ribosome might have made something that was inappropriate. Ribosomes also have something of a lower speed limit. There is no real slow lane when it comes to making proteins. If you're going too slow, the cell will perceive that as being problematic and will try and prevent that ribosome from completing its task. As Barry mentioned in the introduction, a protein also needs to fold. So it has to fold up into a three-dimensional shape in order to carry out its function. And if a protein doesn't do that, the cell perceives that something has gone awry and that it's going to junk that. And then there's other rules as well. RNAs need to be sort of linear, kind of like a piece of spaghetti, rather than folded up into something. They also need to have seals of approval on both ends, and these are commonly called a cap and a tail. And if an RNA doesn't have a cap and a tail, it's perceived as being bad. And lastly, RNAs need to be made inside of the nucleus, which is this structure which houses the genome in the heart of the cell. And the cell has a very low tolerance for anything that breaks these rules. So the cell has an ability to cut up either an RNA or a protein that violates one of these rules. And so it does not hesitate to junk those and anything that fails to adhere to these rules. To help illustrate why these are important, I've highlighted over here that there are examples where cells break these rules or entities break these rules and they give rise to disease. And I put those with a blue D over here. And so I'm going to talk today about this one in particular. This is a major focus of study in my lab, what happens when a ribosome stops too early. And there's plenty of examples of diseases that are caused by cells or genes violating these rules and wreaking havoc on the organism. Also of relevance to our current world is viruses often will attempt to break these rules. And in this sense, you can think of quality control as a first line of defense against invasive things like viruses. So sometimes viruses will have RNA and it will exist in a form that's folded up, and that can be a hallmark of a virus. And so if a cell perceives that RNA is folded up into something, it can then attack it because it might mistake it for a virus. So we study this using what's called a model organism. So our model organism is Caenorhabditis elegans, or C. elegans for short. It is a worm. It's found in soil. It's probably out in your soil right now. If you have a compost bin, it is certainly hanging out in there. They feed on bacteria and microorganisms for a living. And while it may not look very much like a human, at the cellular level, it is very similar to a human. So everything that goes on inside of worm cells is very similar to what goes on inside of human cells. Now, the advantage here is that worms are considerably cheaper than it is to raise an entire human, and also the ethical considerations. So we can grow up millions, tens of thousands of worms and do experiments on them in a very fast time frame, in a time frame that wouldn't be doable for humans. So unfortunately, I can't see you all in person, but I can do a little bit of show and tell because I'm in the lab right now. So this is a plate, and you can see sort of how big it is. It's about the size of one of my eyeballs. And this has on it tens of thousands of worms. So each one of the worms is about 1 millimeter long, and you can't really see it with that, but that's where they live. Give me a sec, so I hooked my phone up to a microscope. I'm gonna log in with my phone and then try and do screen share so that you can see what these things actually look like in the real world. And if you have questions while I'm doing that, just fire off. You can kind of see them now. So this is what they look like. That is right in the middle there.
Oh, yeah.
Is a fully grown adult worm. And then let's see, can I do this? Or is this going to be too problematic?
Yeah. How big is that adult worm?
That is 1 millimeter. Okay, now I'm going to terrorize it. Sorry. Sorry, little guy. So that right there is an adult worm. And then all the little dots around it are its eggs.
Hmm.
And then the really small thing right there that it's crawling towards is a baby. And so it takes about 48 hours for that really small one to become one of those really big ones and then start cranking out more eggs. So by Monday, the small worms on this plate will be the size of big worms, and we'll have tens of thousands of more of them. And that is one of the huge strengths of this system. So we can grow those worms incredibly quickly and do experiments in a much faster time frame than you obviously ethically could with a human or even with something like a mouse.
Mm-hmm.
Do the worms have sex?
For the most part, no. So they're an interesting system. They're mostly hermaphrodites. And that is a really tricky thing to think about initially, because we're used to thinking about male and female species and animals having to have sex in order to reproduce. Because they're hermaphrodites, they fertilize internally, and they can make both sperm and eggs at the same time. One of the real strengths of this system historically has been to study muscle function. And that's for the somewhat obvious once you say it, but difficult to sort of derive from first principles fact that if you can't move, you can't have sex if you have a male and a female species. But if you have a hermaphrodite, because it fertilizes internally, even if it's incapable of moving, it can self-reproduce and then generate more of itself. So actually a lot of what we know about basic muscle function, as far as what molecules are involved, was actually worked out with this animal, not with things like flies or mice. It was generated with this animal for that reason.
So does that make them essentially clones of each other, since they're not mixing chromosomes with anybody?
Yeah, it's really interesting from a sort of species individual level standpoint. These animals on this plate are essentially what's called isogenic, meaning they all have the exact same genome. So their genome's 100 million base pairs, and barring small errors that they make, it's essentially identical down to every single base. For that reason, this has been a really useful system, because you can actually clone them out and isolate several individuals that are exactly identical down to, just about every single atom. That's not the case, obviously, with humans or even with things like flies. So it's been a really powerful system tractably to discover a lot of basic biology, because you can eliminate a lot of the variables of like what causes me to have, lighter hair and my wife to have black hair. And you can't really see it, but there is some bacteria on here that they're munching on. You can kind of see the ripples around, like the waves. It kind of looks like a sand dune. That is indentations in the bacteria. So they're munching as they go along, and they're making ripples in it. And then you can kind of see here, the left side is off the bacteria and the right side is the bacteria. So you can see the left side looks more like a barren desert, and the right side, you see more indentations, more waves.
Do these show up at all in the fossil record?
Oh, that's a good question. I don't think so, because I don't think there's any structures to fossilize. But I actually don't know. I've never heard of fossilized nematodes. There might be evidence, indirect evidence for them, but I don't know of any fossil record for them.
So when you speak of nematodes, is that a general term, or
Yeah.
or C. elegans one type of nematode?
Yep.
Am I
So C.
am I
elegans is
getting
one
that?
type of nematode, and there's all sorts of different species of nematodes. They're all related to each other. They're sort of an interesting branch of the invertebrate lineage. C. elegans just happens to be the one that someone picked out of the compost pile when they were doing. There's other related ones called C. briggsii, C. japonica, so on and so forth. C. elegans has a lot of traits that are really nice, among them hermaphrodism. Not all nematodes even have that.
Okay, my second question is you referred to musculature,
Mhm.
and these nematodes are moving, and you
Yep.
can see that they move. You know, they're not segmented, but what did you mean when you referred to muscle function,
Yeah.
studying?
So they have, they don't, obviously have a bicep like you or I do, but they have along either sides of them, they sort of have what's called body wall muscles, and I'm showing it here as, I can build a simple muscle if I had a pen. Where's a pen? That'll do. If you can see my video, hopefully I won't destroy this. So this is a proxy of what a worm is. Basically a couple muscles stretched out on either side of a pen.
Are you doing a drawing? Because
Let me do a,
can't see that, Josh.
yeah, let's do, I'm gonna,
We have to unpin it.
yeah, I'm gonna stop the worms if everyone's okay with that.
Okay.
All right. And then you should be able to see, give me one sec, let's do, You can see the picture of the worm again? And
Yes,
maybe my video is up on top.
right. You're back to your slides. Yep.
So this that I'm holding up in front of the camera is a pen with a rubber band strapped around it.
Well,
And this is actually, can you see that?
no, we see your slide.
Okay, let's do this.
Barry, everyone will have to unpin the worm picture.
Explain how to do it.
So you go back to gallery view and again click on those double dots and you should get the unpin option.
Okay, so you heard that. Go to gallery view and then go to those three dots and then the windows that show up. Let's see, have to mute. The unpin is your fourth choice. It says unpin video.
Actually, I might be able to do this by,
Okay, and that worked. Okay.
that may force it.
Thank you, Brian.
Again, you can't do your own tech support.
Yeah. So
Can you see me? Am I appearing on people's
Yes.
screen?
Yes.
Yes, I can see.
The question was about muscle and what muscles does a worm have? This is basically a crude model of what the worm is. So it's a pen and I've strapped a rubber band on both sides.
let me just interrupt, Josh.
Yeah.
So if you had to go to gallery view to unpin and then go to speaker view, Click on speaker view, and now you can see Josh. Full screen. Go ahead.
And the worm has two sets of muscles, one that's on one side of its body, another that's on the other side of the body. And when you're looking at it from the top down, it kind of looks like this. And the way the worm moves is by basically flexing this half and then flexing this half and going back and forth. And by flexing those two muscles, it can sort of wiggle. And by flexing up here, it can move its head. By flexing back here, it can move its tail. So that's how, but you don't need those muscles on either side. That doesn't have anything to do with its reproductive system. And so it can still be reproducing internally, and these muscles can be completely defunct, and it'll still be fine. It'll just lie there on the plate, flaccid. So that's one example of how they've been used to study muscle function. They also actually use their mouths to pump too. So you can study muscles in their mouth as well. Now, I hope my slides are back up.
It's good.
Perfect. So that's, a brief little description of why we know and love this organism. A lot of basic biology has been worked out in this organism. As Barry alluded to, my postdoc advisor won a fancy prize for biology that he discovered in this organism, which actually is also biology that's true in our cells as well. It's also very economical, which is nice when you're working at a public university. So, that plate that I showed you of the animals, I don't know, costs $1 or something to make. And so you can grow up, hundreds of those plates, and you can hand them out to your students, and your students can play around with them and learn how to manipulate the system without breaking the bank, which is nice. And another really nice feature of this, going back to the sort of what I study, is that you can actually break their quality control processes and they still live. So if you break a lot of quality control in humans, we die. It's embryonic lethal. And so it's very difficult to study with humans or with any sort of mammal for that reason. But a worm can get by without its quality control processes. And that allows us to do experiments that we couldn't do in other animals. So here's an example of a experiment that we do. We can grow up a large number of animals, worms, and then we can engineer them in a way where they will light up if their quality control processes are compromised. So they'll literally start just producing light, in this case, glowing green. So we can grow up hundreds of thousands, millions of these animals, and then look for a rare animal that suddenly lights up. And we know that that animal is broken in some way that's interesting to us. That's an example of a suppressor mutant. And we've got a freezer full of suppressor mutants that are all broken in various ways pertaining to quality control. And we use that to help identify how that quality control works. Here's an example of what these actually look like. So these are some strains that were made in my lab that were engineered to fluoresce green when their quality control processes are compromised. And so starting up here, you can sort of ignore the names. The names is sort of boring genetic nomenclature for C. elegans. But the starting strain doesn't really glow at all, but you can make mutations that cause them to glow. And that's shown here as well. There's a bit of a gruesome aspect of these animals, which is, as I mentioned, they are hermaphrodites, and they are capable of fertilizing internally. Sometimes, if the mother does not succeed in laying the progeny fast enough, they will actually hatch inside of her. And it's pretty gruesome, just like a horror movie, Alien. They can actually hatch inside, and eventually they'll crawl their way out. Sorry to spoil some of your breakfasts. It is a sort of it's kind of humorous for us worm people, because I don't know, it's the first time you see it, it's horrible, and then the second time you see it, it's like, oh, and then it's sort of just a running joke amongst worm people.
Does that kill the mother?
Eventually, yes. Yeah. But it's a trade-off that's, while it doesn't seem like it's in her interest, it's in her genetic interest, because she sacrifices herself, and then these 10 animals get to survive. And they're 10 animals that are all directly from her genetic material. And so from a genetic perspective, it's a great idea. It's not a great idea as far as her being passes on rather quickly. But she gets 10 clones of herself. And from a genetic perspective, that's a great idea. Other questions?
What is her lifetime?
It depends. Generally, these animals live for only about 2 weeks. There's some variability, you know, depending upon their environment, but most of them don't make it past 3 weeks. Some of them will poop out after a week and a half. They're reproductively competent in about 48 hours, which again goes back to the fast generation time. So those worms that I showed you, the small ones, will be cranking out eggs by Monday. But by December, they'll all be toast.
Can you tell who is who? Do you tag them in any way?
You can. There's a couple ways of doing that. One is genetically. So if you've, because animals on a plate are essentially exactly the same as one another, you can introduce an animal that's marked in somehow that's different from all of those, and then you can pull it back out if you need to. You have to mark it usually genetically. You can, it's really difficult to actually like make a mark on the animal, like use a laser to ablate some cell or something. You can do that, but that's much more difficult and is beyond me, to be honest.
Thank you.
How many rounds of egg laying does an individual make?
About 300 to 400. So one hermaphrodite gives rise to 300 to 400 eggs, and each of those can give rise to 300 to 400. So that's really nice, because if you have one worm that's really precious for some reason, you can put it on a plate and it will clone itself 300 times within 48 hours. And then you've got all sorts of versions of it that you can then study. And so it's really nice for mutations and screens like what we do, because if you find one worm that's incredibly precious, it will clone itself basically overnight.
What is the mechanism for dying? I mean, what happens? What doesn't work?
That is a great question. It's an area that I know a little about, but I think we are collectively still learning about it. There is a signal that goes throughout the entire organism when it does die, or like the split second right before it dies. That is some small molecule of some kind. We don't yet know where that signal comes from or what exactly the purpose of that signal is, but it's actually, it causes them to glow blue for a split second. So it's kind of, I don't know, you know, what that means spiritually, but there's this split second right before they die where the whole thing will just glow blue, and it's called death fluorescence. It is associated with the process of them dying, and it is an organism-wide signal. Last I read about that was 2015, and to be honest, I haven't kept up on that literature. But there is a organism dying process.
Joshua, you used the term several times. If we find one that's incredibly precious, what do you mean by incredibly precious?
Yeah, so, you know, this plate that I showed you has, you know, 10,000 animals on it. We have plates that are the size of my head that might have hundreds of thousands or millions of animals on them. Sometimes we find one worm on there that we need for some reason. And it could be, for example, a worm that now glows green that's in this background. that worm presumably glows green because something has been altered about its quality control machinery. We would like to study that. And so you want to get that worm isolated. And and the sort of term that we use is that worm's incredibly precious, because if you lose it, you don't know what was wrong with it. We would like to study what was wrong with it and what went wrong with it. And so you might have, you know, quite literally a needle in a haystack, but it's a needle that you can put on a plate and will clone itself 100 times. Yeah.
Why
Yeah.
haven't they taken over the earth if they're so good at reproducing?
They're good at reproducing, but once they run out of E. coli, the gig is up. So they have to eat for a living, and they need to eat E. coli for a living. And so when E. coli runs out, they go into a starvation mode and they stop their development. There's actually this whole study of, there's an alternate form of development that they can go through called Dour. And the Dour form is sort of like the tank version of a C. elegans. It has a much harder outer shell, and it can survive even rough chemical treatments. And it can live even for months. So it's, you know, someone asked me how long do they live for? They typically live for a couple weeks, but Dour, which is a specialized form, can live for a couple months, and it's basically designed to like hunker down and wait for E. coli to come back.
They have
Since we have E. coli in our gut, I think, do these worms live inside humans also?
They don't, but there are versions of nematodes that do parasitize humans. So they're the basis of a lot of diseases that predominantly affect tropical areas. For example, I think elephantitis is based on the growth of a nematode, Brugia malay, that parasitizes humans. For the most part, they don't. These ones are benign, which is another reason why I can safely hand them out to undergrads. Luis, you had a question?
Yes. What do you do with these worms?
What do I do with these worms? So we use them to study quality control, and we use them to try and identify the machinery that actually does that quality control. So I sort of use the analogy of, you know, a car coming off an assembly line, and like someone doing quality control can look at the car and be like, yeah, no, that brake doesn't work. Like, and that's pretty obvious. But what the cell has to do is it has to basically feel up the molecules as it makes them and say, yeah, no, that feels about right, or that doesn't feel about right. And we don't actually know what that hand is that's essentially grabbing onto the molecules as they come off the assembly line to check them. And so an example of work that just did that was from a PhD student in my lab, Marissa, and she actually used these animals right here to discover one of those genes that was required for doing that. And so she got to name that gene as well, and she called it Nonu1 because she thought that was a cute name. There's a molecular biology story behind why she thought Nonu was a good name, but she got to name it because she discovered that gene. And this turns out Nonu is a gene which is conserved between worms and humans and yeast. And so it's a very ancient protein that no one had discovered before, but she found because she found one very precious worm that happened to be a mutant for that factor. And then she was able to show that that factor was sort of one of the things that was grabbing onto the ribosome as it was trying to translate things.
Yeah. Josh, it might be worth mentioning that even though we know how many genes all these creatures have, we don't know the function of most of these genes.
Oh, yeah.
We're still very much in the stage of trying to figure out what all those genes do.
Yeah, yeah. And so, you know, the worm is a great system for that. You know, so we have humans, we have 25,000 or so genes, and we know what a fair number of them do, but we don't know what all of them do. And this was an example, this gene that my student Marissa found was a gene that was known to exist, but no one could actually figure out what it did before she sort of discovered its function. And it's known to exist, you know, in all eukaryotes, so going all the way back to yeast, worms, humans, all of us. And she finally was able to ascribe a function to it, but there's still, you know, I don't actually know what percent of genes we have no idea on. Barry, do you know that? Is it like 10, 20%?
Well, one of the most interesting numbers I heard is that E. coli bacterium, which is simple and probably one of the best understood organisms in the world, has 6,000 genes. And as of a few years ago, we only knew the function of 3,000 of those genes.
Hmm.
So it's generally at least half unknown.
So that gene that produces a protein that does the check.
Yeah.
And so you figure there's probably like a dozen or more of those, or how many of those have you discovered?
Yeah. So we've got about a dozen or so acting. So it depends on which of these sort of rules that you're talking about. So each one of these rules has a different set of effectors that carry it out. And so Marissa's gene that she found was actually involved with rule number 4 here of ribosomes moving too slowly. And we have about 3 or 4 genes that we know act in this pathway. We've got 7 to 9 that act up in this pathway up here. We don't know any of them that act in this pathway yet. That's a big unknown. We know about 4 to 5 of the ones that act in this pathway, that act in this, in enforcing this rule. And there is some overlap between them, but there is also some distinctness. So some of the machinery that carries out this, enforces this rule, is actually different from the machinery that carries out this rule. All right. So what I want to talk about in the last bit here is actually rule number 1, which relates to what happens if a ribosome finishes its job too soon. And this is a substantial focus of my lab and also a big tie-in with disease. So again, shamelessly stealing from Professor Bowman's slides. So you have DNA up here, which is A, T, C, and G, and that gets copied into RNA, and the T gets swapped out for U, and then that gets translated into a protein here. So what happens if you have a mutation in here that changes the nature of this information? So if you change this one base here, you can kind of see this G goes to a U. That actually changes the meaning of this protein. So UAG can be interpreted as a stop signal, just like how TAA can be interpreted as a stop. And so when that happens, when the ribosome translates this, it terminates right here. And so you only have half of the protein getting made. So you have one very simple error. You just changed one letter to another letter, and it basically cut off the entire latter half of this protein. You no longer get to make that protein. It's not really an academic consideration. So when molecular biology came around and we started applying those techniques to human patients, we discovered that there were people walking around that had mutations like that. So this is an abstract from a paper, and based upon the molecular biology that Barry's taught you so far, I think you can understand this abstract. So this was published in 1979, and this is one of the first instances of someone walking into a clinic and very obviously being the result of genetic disease. So the abstract reads, we determined the complete sequence of the 5' region of the first amino acids of the beta globin mRNA. And so there's a gene that encodes for the beta globin mRNA in a patient with beta thalassemia. So this is someone who walked into the clinic and had beta thalassemia, and they decided to get the DNA sequence of their gene.
Explain beta thalassemia.
Oh, does anyone here know or have beta thalassemia and would be comfortable sharing that? So it's a deficiency in one of your hemoglobin subunits that causes you to not be able to carry oxygen as efficiently. And so this is someone who presented as not being able to carry oxygen, and they decided to look at their gene. And so they identified the defect as a single nucleotide substitution in the coding region of the beta globin gene. And so it turned out that this person had a mutation like this, which caused one of those stop signals to happen too soon. And as time has gone on, we've sequenced more and more people, thousands of genomes at this point. Everyone, on this Zoom call, myself included, has some of these types of mutations lurking in our genome. And over the years, we've identified around 1 in 10 or so genetic diseases are caused by mutations like this. And so this is a major pathway of focus for the lab, because we're interested in understanding what the consequences are for the cell when this happens.
And why are they called nonsense mutations?
They're called nonsense because of, because this used to be called nonsense. a stop signal used to be called a nonsense signal, because each of these makes sense to the cell, because the cell can make sense of it and read it out as an amino acid and make a protein from it. But something like TAA does not make sense to the cell because it can't read it out as an amino acid, so it's nonsense. So it's kind of a historical thing. They were originally identified as a kind of mutation that happened in bacteria, actually, until it was realized that nonsense happens all the time. All sorts of nonsense-related puns in the lab related to that. So there's two things that happen. The first is relatively obvious, which is that because you have nonsense, or you have a stop signal, you no longer get to make the second half of this protein. So you've only got half of a machine. That's a potentially really bad thing. So imagine you made a car, but you left off the brake, or you left off the steering wheel, or you made a door and you never bothered to give it a handle. Those sorts of machines are kind of the stuff of nightmares. And so the cell doesn't want you to be making half of something. It says you have to finish the entire job or you get nothing. Because in the analogy, it's better to have no car than it is to put a car out on the road, give it a gas, and not give it a brake. And so one of the things that the cell does is if it determines that a protein is only half of a protein, it will actually start attacking the mRNA and get rid of that, because again, in the analogy, it's better to not have a car than to have 100 cars on the road and give one of them no brake. So that's a very fundamental principle, and that's true of all of our cells. So, and this is true of worms, this is true of humans as well. And so a substantial focus of my lab is studying how the cell identifies nonsense and how it decides to attack that mRNA.
So if you replace the protein that's only half made in the cases of the people that had that deficiency you were talking about, does that correct their deficiency?
Sometimes, yes. And so that's the million-dollar question. So, going back to this person right here who walks into a clinic and can't carry oxygen in their cells through their veins, that's for two reasons. One, they only have half of a beta globin, but also their beta globin protein is actually dampened down. So you might be able to help them if you could give them back their half a beta globin. That might be better than having no beta globin. And there are some pretty clear examples where that's the case. And so one of the hopes of this line of research is that if we could understand how the mRNA gets attacked, we may be able to shut that down in some genetic diseases and potentially give those patients some reprieve from the genetic disease that they're suffering from.
Josh, I have a question.
Yeah.
So this could be caused by a mistake in the DNA, and in that paper, that person did in fact have a DNA mutation. Isn't that correct?
Yeah, yeah.
I assume that where this happens at a much higher rate is when the RNA is synthesized, the mistake is made. And so there's a small number of these being made kind of at a low level in the background. Is that correct?
Yeah, that is. It's about 1 in a million at the level of RNA, which sounds low, but when you think that there's tens of thousands of RNA molecules in a cell at a given time, and each one of those is 1,000, you've got tens of millions of bases. So you've got about 10 mRNAs per cell that have a mistake like this. Yeah, so it's pretty, and that's every single one of our cells. So. So that is the bulk of what I wanted to talk about today. I'm going to jump down to, these are the people who actually do this work while I sit in my office and talk to you all and teach students and so on and so forth. This is the lab, this is our lab meeting, which of course has to happen on Zoom these days. This is Marissa, the one who looks like a ninja. This was her in lab the other day. She has to wear a face mask, obviously. This was the one who discovered that new gene, the one that she got to name Nonu. And then these are some other PhD students, an undergrad and a tech. These are organizations that provide money for us to do this. Amongst them are the NIH, paid for by your tax dollars. So thank you all. Your tax dollars contribute to research like what you're seeing here.
Okay. So another question would be, in your paper, the gene that was identified is an endonuclease. So you might explain what this gene does.
Yeah. So the gene that Marissa found, this one, Nonu, that she got to name, is a class of gene that's known to attack and cut RNA. So it's called an endonuclease. And what we think it actually does is binds inside of the ribosome and then cuts the RNA. And that elicits its own host of red flags for the cell, because the cell says, you're trying to translate something which has no stop signal. That's a really bad idea. And that's in response to the ribosome moving too slow. So you can kind of imagine that if you're not doing this fast enough, if you're not shunting these things through and trying to make peptide bonds too quickly enough, Marissa's gene, Nonu1, hops in here and says, nah, that's too slow, you're done. Finish off with it.
Okay, other questions for Josh?
Luis, I can see you.
That's, yeah. Would you mind if I took a picture of that cell? I think that's a fascinating picture.
Which one?
The one that you started with.
Oh, yeah. Yeah.
The multicolor cell.
That one?
Yeah, I've never seen a picture like that. I really love it.
Yeah, so if you Google this person and then probably molecular biology, you'll find all sorts of examples of this. So this is, so now that I've talked a little bit about the molecular biology, this kind of light blue spaghetti that's coming out of here is an RNA.
Okay.
And this is leaving the nucleus. So this is the DNA in here. And the RNA is getting moved out. And then this is his depiction of a ribosome, which is trying to translate the RNA and make a protein from it.
Where's that?
Josh, I have a question. when you say the process is going too slow, how do you measure, there's a concept of time and progress per
Yeah.
time. So how is that measurement?
Yeah, that is a great question. And it took people 11 years to answer that. So how do you measure speed? You know, is there, you know, there's no radar in the cell, right? It's not like, you know, everything's going down the highway and you're just trying to measure. So it turns out, and this was work that was done by Hani Zeher, who's an assistant professor at WashU in St. Louis. He came up with a really cool idea, which is speed is measured by traffic jams. If you have a traffic jam, you know that things are going too slow. And if you don't have a traffic jam, who cares? And that was the idea that he came up with. And it turns out that's right. And so basically what happens is so you have an RNA, like this RNA, or this RNA, and it can actually be translated by 2 ribosomes at the same time. So one ribosome can hop on, start making protein, and another ribosome can hop on, start making protein. And what Hani found was that if that first ribosome goes too slow, you get a collision. And it's like bumper cars. And it's actually that collision of having a traffic jam that the cell uses as the red flag. And that's where MRSA's protein binds, it turns out, is it actually binds at the interface of 2 collided ribosomes. And so that's how you can sense speed. It's indirect.
A question I have about that is, so I guess one kind of mutation you could get too is to get these relatively rare codons. That would, as I understand, that's one way to slow down the speed at which things move along, because some 3-letter codes are abundant and some are rare, and the rare ones have few tRNAs to recognize them. Is that a part of the story?
Yeah, that's how we elicit traffic jams in the cell. So we give them a run of codons that are very rare, that they shouldn't be able to translate quickly. Analogy, it's sort of like, I don't know, giving them, you know, 10 speed bumps in a row on the freeway. If they're going into if the molecule's going into that at, you know, 100 miles an hour, by the time it hits the 3rd speed bump, it's going to be complete chaos. And so that's how we elicit that problem.
In looking up, you know, introduction for your intro, I saw that there are some specific diseases that you're focusing on that are related. What are those specific diseases?
There are diseases that are like this one, where actually, like this one. I'm interested in situations where you have an early stop signal that causes loss of the RNA, even though the protein is perfectly fine. And that's a minority of all what's called nonsense diseases. So it's a minority of this 11%. But it is a area where if we could find some way of shutting down the attack on the mRNA, we might be able to offer some reprieve to those patients.
So I have a question about something you just said a moment ago. You said that you find the traffic jams because you have two proteins being created at the same time through different ribosomes, but they're following the same piece of RNA through. Is that normal to have that more than one at a time being produced?
Yeah, we're very good at multitasking on the cellular level. you can have 10, 20 ribosomes all stacked up competing on the same bit of RNA trying to churn out protein. That is one advantage of this system being this way. So you have one copy of your genome, you can make 10 hundreds of copies of your RNA, and each one of those RNAs can be bound by several ribosomes, tens of ribosomes, and you can potentially get thousands or tens of thousands of copies of protein. So it allows for an amplification to get a lot of protein. And things like myosin, what we have in our muscles that allows us to flex, that undergoes a huge amplification like this. You still only have one myosin gene, but because you have so many RNA molecules and so many proteins made from each of those, you get a huge amplification of the amount of that gene and allows you to get, protein to where you can actually build an entire muscle, makes up your entire arm off of that.
Okay.
So it just makes as much as it can always, or is there some place where it stops it because it doesn't really want all that much, whatever it's making?
Yeah, and that's an area of study called gene regulation. And that was actually what I did a lot of my PhD work in. So how does a cell know how much of a given protein to make? You need to make a lot of myosin because you need to build an entire muscle, and that's going to be most of the cell. But something like Marissa's protein nonu, you don't actually need that much of nonu, because you don't need one molecule of nonu for every myosin. You only need one per ribosome or so. So there's a whole field that's focused on understanding how the cell does that. And it turns out there's a lot of waste, and a lot of the excess that gets made actually gets funneled through quality control pathways.
Thank you.
Yeah.
So Josh, you also, I understand, use the CRISPR technique quite a bit in your lab. And this is, of course, something we're all hearing about now. So you might just say a little bit about CRISPR and how you use it to study worms.
CRISPR was also, this is a little bit of history. So in science, we refer to our, some people refer to their lineage of like who they got trained by. So like, Barry mentioned that I was trained by Andy Fire in my postdoc. So the science nomenclature for that is sort of, Andy would be my father scientifically because he trained me. So Jennifer Doudna is the scientist who won the Nobel Prize this year for discovering CRISPR. She is, in scientific terms, my grandmother because she trained my PhD advisor. So it's a little bit of science culture there. So let's show, actually, we'll stick with this one. So one of the things so CRISPR allows you to edit the DNA in a cell, and that's very powerful for the kinds of things that we do, because we can create mutations like this, and we can create them wherever we want. So if we wanted to study this process of nonsense, we could use the CRISPR system to make a cut here and then offer the cell a different version that hard codes a stop signal right here. That allows us to engineer the worms to have a mutation at an exact site that we want it to. That's actually what went into engineering these worms right here. We engineered them to have a specific lesion, and then we also engineered them with a protein that lights up green. And so normally they attack that because they perceive it as it being a lesion or as it being problematic because it violates the rules of quality control. But by breaking something like nonu, they lose the ability to attack that, and then they start making the green protein. So we use CRISPR daily. It's actually what I was doing this morning. Before this, I was sitting at the microscope trying to put CRISPR into some worms to make a mutation at a specific protein that we study that I didn't talk about today.
And when you put it in, you actually inject it, is that right?
Yeah, we inject it. So you can kind of see their structure here. So actually this little blob right here, this little series of blobs, these circles, are the oocytes. So the eggs developing inside of the mother. And they pass through the spermatheca right here. And then they start undergoing development, and then they get laid through this opening right here, the vulva. This whole arm of the animal is essentially an egg producing machine. And we can, using a very fine needle, inject DNA into there to do something molecular biology wise. And so what I spent this morning doing was actually, I had a needle hooked up to a microscope, and I inject 25 worms that look like this worm, each with a mix of components to do the CRISPR. And some of those will get taken up into the DNA of the cells and cause mutations. And today is Saturday, so 48 hours, Monday, Tuesday, I'll be able to start looking at those animals.
Okay, any further questions for Josh here? I'm going to stop your screen share, put everybody on, so I can see gallery view. There's all the, any further questions? Further questions? Okay, I'm not seeing any. Well,
wait, a hand from Mark, was that? Or is that just saying hi?
oh, okay. Oh, they were applauding.
Oh.
Oh, okay. All right, silent applause here. Well, thank you very much for spending your Saturday morning with us, Josh. And I can kind of see out the window here. The waves do not look very good this morning. Is there a good swell?
2 o'clock, there's supposed to be decent surf. There's a low tide around 2 or 3. So.
Okay, okay. Well, that's good to know. All right. Well, thank you. And so next week, our speaker is going to be Martha Zuniga. Martha is an immunologist. You know, this coronavirus pandemic is in many ways all about the immune system. And she's going to talk about how our immune system recognizes viruses, and also about how the fact that our immune system can be turned on to too high a level such that it actually starts to attack our cells, and that's a serious problem in some people who get infected with the coronavirus. So we'll hope to see you all again next Saturday, and we will have lecture 3. So thank you very much, Josh. All right, and we will see you all in a week.
Thank you, Josh.
Sure, thank you all.
You know, it was fun being in your lab.
Yeah, you can kind of do a field trip.
Yes, thank you.
Okay.
Thank you, Barry.
Okay, bye, Josh.
Always want to talk.
Mm-hmm.
Have you stopped recording?